Hello and welcome to this podcast with Sino Biological. My name's Will Suter. Today we'll be talking with Dr. Yuning Chen, who is the R&D manager at Sino Biological, and he'll be telling us about Sino's response to the COVID-19 pandemic, how they were able to rapidly pivot to focus their efforts on the virus, and how their expertise in recombinant proteins has allowed them to contribute to the global effort to develop vaccines for COVID-19. Hello, Yuning. Thank you for joining us today.
Hey guys, how are you?
Yeah, great, thanks. So to start off with, would you just give us an introduction to yourself and your role at Sino Biological?
Yeah, of course. So I'm a research and development manager at Sino Biological. I have a background in protein biochemistry. I got my PhD from Ohio University back in 2012. And then I did a few years as a postdoc working at Penn State. And my research focused on plant-based proteins and polysaccharides. Then in 2016, I went back to China, joined a small biotech company, worked there for 2.5 years. My work was focusing on antibody-related research and development. And then I joined Sino in March of 2019. And Sino here, I'm in charge of recombinant protein design and also monitoring the recombinant protein expression and development platforms. So yeah, still a lot of work surrounding recombinant protein technique.
So could you talk a little bit about Sino Biological? What does the company focus on? What are the kind of specialties that it brings to the market?
Okay. Well, I can give you guys a short, one-sentence summary. So basically, we are a biotech company, and we devote our resources in developing high-quality recombinant proteins and antibodies. And our products are mainly used for the support of research and development for like a field of life science and drug development. So the company, I believe, was established in 2007, and our CEO, he is MIT laureate. His specialty is recombinant protein expression using mammalian cells. So this is basically the cornerstone, the technical cornerstone on which the company is based. So we do a lot of product development using this mammalian cell recombinant protein expression platform. When time goes on, we're becoming more and more comprehensive in terms of recombinant protein expression, and we integrated different platforms such as insect cells and E. coli and yeast. So we have built a rather comprehensive pipeline in this regard. We also incorporated the team that are specialized in antibody development. So basically, I think at this point, our portfolio can cover from antigen discovery all the way to antibody development. So it's pretty comprehensive in terms of biotech technology on this regard.
So we're going to be talking today mostly about COVID-19. Obviously, that's been a huge topic of discussion for lots of reasons recently. But how did Sino Biological operate within infectious disease research before the pandemic hit?
Basically, our main technical platform is recombinant protein and antibody development. So we use this platform to, develop reagents against different infectious diseases that are known to human beings. So for instance, we have a large collection of recombinant proteins from, influenza or HIV or Ebola. It's kind of like developing something that we have. So when the time comes, these are needed to be deployed in any shape or format so that to move the research of infectious disease forward as quickly as possible. Because I think the time when either outbreak or a pandemic happens, time is the key. If we can have something quick and high quality reagents as quickly as possible, it will inevitably help us deal with pandemics. So I think one of our aspects is to using the recombinant protein expression technique to prepare these so-called viral protein antigens or bacterial protein antigens. And on the other side, to make these proteins are actually quite tricky. First of all, it depends on what kind of virus they're derived from, and each protein has its own unique characteristics. So even though we do have several very well established recombinant protein expression platforms, but we would still need some experience in terms of how to make different proteins from different viruses or even different variants of proteins from the same virus. So I think before the pandemic, before COVID-19, a lot of work that we did, a lot of resources that we invested in this sort of platform establishment so that we can try to express as many recombinant viral protein or bacterial protein as possible for us to, just familiarize with, what we're dealing with, and also get to learn some tricks, if you will, along the way to so that we can be prepared for future pandemics. And well, unfortunately, we do have one at our hand. Another thing that we did mostly was to developing those quote unquote stock antibodies. And these antibodies can be deployed rather quickly for immunological assay development. So once a virus that it has been identified, which is maybe similar to a previous virus. So in this case, the COVID-19 is kind of similar to the SARS outbreak happened about like a decade ago. So these antibodies really come in handy, and we can just use these antibodies, especially during the beginning of the pandemic, to either develop some quick serological assays or to help companies that are for vaccine development to use these antibodies as detection reagent for them, so they can assess the efficacy of their vaccines and to see if their vaccines are really working or not. So before the pandemic, this is pretty much what we do. I think we can call it like a preparation, but I would say it's more of a like a learning curve. So we're trying to peek into the protein biology of the viruses and to try to understand them as much as we can.
Great. Yeah, it sounds like you're about as prepared as anyone could have been. So when the pandemic did hit, how did that impact your research process and your product development kind of pipeline?
I guess one of the major impacts that we encountered was, or and still is, like the timeline. Everything is being squeezed into a very compact timeline, and because the virus is changing all the time, and we have to be able to catch up with it, right? So besides the development of these wild type proteins, we're constantly trying to make these mutated variant proteins from the virus. So we're really using our platforms, working it really hard to try to make as many proteins as possible. And because the customers usually starting to require a significant quantity of the proteins, the impact is on both the depth and the width of the platform. So we're kind of like stretching it very thin. On the other hand, we have a quote unquote high throughput recombinant antibody expression platform with which we constantly try to create recombinant antibody libraries for clients. And due to the emergency of this pandemic, and also due to the characteristics of the variants, especially the receptor binding domain of the SARS-CoV-2 spike protein, which is, I think, the main focus of many researches and drug development efforts. Even though the RBD, or the receptor binding domain, changes constantly, but the changes will mainly occur in one amino acid or maybe a few amino acids. So it's kind of like antibody expression. So the changes in different antibodies lie in their CDR regions. So we can build a template of an antibody, and by changing the CDR template, we can quickly generate a library of vectors for antibody expressions. So this is kind of similar for the case of the SARS-CoV-2 variant. So we also built like a template for the SARS-CoV-2 spike protein or segments of the spike protein. And whenever a variant or several variants comes our way, we can quickly produce these proteins in the form of recombinant proteins and supply them all over the world. So there are all kind of problems. The world has been hit with something so massive. We've never experienced something like that in our lifetime. So the research has changed significantly, and also other aspects of life, for instance, like the logistics side of the things have been changed as well. We also had to devote some of our effort into dealing with the customs and all these kind of product development related topics, but not necessarily directly involved with product development. So yeah, we've been coping and trying to catch up with the world as it goes.
And how about life in general? How has the company adapted to quarantine requirements, remote working, things like that, and travel restrictions? Obviously, there's some kinds of work that can be done quite effectively from home, but you can't really take your lab home with you, I'm guessing. So how have you coped with that?
Yeah, well, I mean, you're right about that. So we really don't have the luxury of working at home, and because of the pandemic and because of all the restrictions, things have been really tough, but the workload has been, I think, at least doubled or sometimes even tripled. And, because of all the, travel restrictions, we have to cancel like basically everything. People in the US, they can't go home for Christmas or Thanksgiving. In China, the New Year was, quote unquote, canceled. So can't go anywhere and can't see our families. And it was really, I think it was really tough on the emotional side of people as well. But I think we did manage to, endure this hardship and making adaptations as much as we can. Interestingly, some of our workers, they either live in some, high risk area or they've been to some high risk area. When they came back, they have to be quarantined for 14 days. There was a time there was always someone missing. Okay, then after 2 weeks they come back. But as you mentioned earlier, we can't really take the labs with us, so just try to work with quote unquote limited personnel and try to get the work done. But I think our lab technicians and the people who does the work and do all the experiments, I think there is a real hero. They make all this happen, so I can sit here and talk to you guys about it. Yeah, I can't do anything without them.
Yeah, no, it definitely sounds like it's been tough, but I'm sure you've done a really great job sort of keeping going through it all. Going back to variants, as you were talking about, obviously at the moment that's a sort of big topic of discussion with the sort of Delta variant causing new waves almost everywhere, and more variants sort of looking like they're on the horizon as well. How do you kind of keep track of the developments in new variants and where they're coming from? How does your kind of approach with recombinant proteins allow you to respond to that?
I think in order for a variant to be established and to have a certain prevalence, it requires some time in order to compete out its peers, essentially. We constantly monitoring, a few reliable sources, among which are CDC and the WHO. They constantly post these, quote unquote variants of concern, and they document, what kind of changes are there in these variants, and especially, I think these, variants of concern are all, named or categorized based on the spike protein. And so that was also our, main focus on, making these recombinant spike proteins as well. There is also I think a website or a database called the GSAID, which is kind of like an open source, so where people kind of deposit a lot of sequenced information into this website. So we have actually established a quite extensive data bank for this virus as well. This database, the GSAID database, is very useful for us to obtain actual sequences. With the combine of these three resources, we can identify the variants of concern, and we can identify the mutations occurring on these variants of concern, and we can convert these information into recombinant variant proteins as quickly as possible. So that's for the spike protein, of course. But for the nucleic capsid protein, things a little differently, because for one thing, it doesn't change so much. But when it does change, people don't tend to track the changes as actively as that of the spike protein, because the spike protein is where the virus interact with the host cells and try to do its magic to get in. And once there is a change, it can sometimes throw the antibody protection out of the window, and sometimes it can also compromise the efficacy of these therapeutic antibodies. But the nucleic capsid protein does not do those things. It's basically there to stabilize the virus genome, but it's also important because it's becoming more and more obvious choice of developing those serological assays to quickly identify if someone has COVID or not. So with the help of GSAID, we can quickly we're constantly doing database mining, essentially just trying to identify those like high frequency mutations of this nucleic capsid protein and different versions of this protein accordingly, and then to test the antibodies that we have raised against this particular protein to see if these mutations would cause the compromise in their efficacy, and to essentially pick out a quote unquote broad spectrum antibody pairs or broad spectrum antibody. So to, essentially to deal with all different types of mutations so that these antibodies can be used to form a relatively broad spectrum serological assay to see, which one the people with the COVID.
So let's talk about vaccines a little bit. So
Sure.
obviously the sort of end goal of a lot of this research is to try and develop vaccines, improve vaccines, and make it so that although you can't obviously predict every single variant that's going to emerge, like the ideal is that you want a vaccine that will protect against all of them, right? So
Mhm.
how does the recombinant protein approach help you kind of get there, and what are the challenges with kind of trying to predict what those changes in the variants are going to be and how you can sort of anticipate them in the vaccine development?
So this is a very hot topic, kind of like the core concept is to develop kind of like a universal vaccine for, in this case, for COVID and different variants. And I think people are trying to develop such vaccines for different viruses as well. I think most recently there was a clinical trial for a universal flu vaccine that has just entered phase 1 clinical trial. The core concept of this, universal vaccine is to, like you said, is to be able to recognize or elicitate the body to generate antibodies that can basically recognize and neutralize different variants of the protein. Because the proteins are changing, but lucky for us, in terms of COVID-19, the protein does change, and it does change at a relatively high frequency because it's an RNA virus. But the changes are not significant. It's not going to just throw the current antibodies, throw their efficacies all the way off, but maybe to reduce their efficacy because certain epitopes on the spike protein is changed. One approach would be to carefully study the core structure of the protein, to target those regions where the mutations do occur, unfortunately, but they occur at a lower frequency, basically to identify a region that is quote unquote conserved between different variants. So that could be a little bit challenging, but I think in the case of the SARS-CoV-2 virus, S2 region of the spike protein is relatively more stable. I think people might focus some of their efforts on this region in order to develop a, quote unquote, universal vaccine, maybe for SARS-CoV-2 virus. So the variants occur, the virus mutate, but that's fine. Like I mentioned earlier, the mutations do occur, but they occur at a few amino acids at a time. So I think it would make sense to, if we can, solve the problem by using only one recombinant protein, or no matter it's, served as a recombinant protein format of vaccine or an mRNA form of vaccine. But if we can't do that, we can't, solve the problem using, one candidate, one protein. Maybe we will be able to use several proteins to make it into a combo so that we can cover as many epitopes as possible and to cover as many changes as possible. And I think this is also the basis for the current universal flu vaccine. So I think that would also be maybe an approach that we can take for this kind of work.
Yeah, that's really interesting. I heard a suggestion recently that with the spike protein in particular, because it's the mechanism that the virus uses to infect cells, if there are sufficient changes to the spike protein structure to where it would kind of throw off some of the vaccines and sort of reduce their efficacy, it would also actually just make the virus itself less effective at infecting cells and make it less infectious. So there's kind of a trade-off with the regions that are being focused on. If they do change, they actually give you more time because that actual variant would be less infectious. But yeah, it's definitely interesting to hear how some of this research that's kind of being, as you say, in the emergency of COVID, it's kind of being pushed forward and kind of these approaches are being developed, how they're going to eventually sort of feed into, other vaccines, other viruses as well. So what would you say the impact has been, overall of COVID-19 on the landscape of virus-related biological reagent development? And what do you think the future would hold for future outbreaks? If there are, other pandemics or other sort of viral epidemics in the future, do you think the general sort of response will be better because we've had this pandemic now and everyone's sort of developed the methods to deal with it better?
They always say if you want to, unite the human race, you have to give them a common enemy. And I think this time nature does offer us a common enemy. And I mean, our responses are, unprecedented. I think at least for the for the scientific community, we do see a unprecedented global effort for collaborations. Once the virus has been identified and its genome was sequenced and published, and made available worldwide, and then kicks off all the work of vaccine development, or for us, protein and antibody development as well. But everything was about sharing information and all the research you can see. I look at the preprint on a daily basis in order to track the progresses that we're making, both in terms of therapeutics development, and also in terms of to understand the biology of this virus. But this preprint platform does give us essentially highways for all the information is available, even though the papers that are put forward on these servers are not peer reviewed, or at least not has been peer reviewed. But the information presented are useful for us to get understanding and to inspire maybe like other researchers, or to promote collaborations between different groups, or even between different countries to solve this problem altogether. And also, I think we're seeing like the embracement of new technology, and I think the most obvious one has been the use of those like RNA vaccines. They've never been used before, and for COVID, this is the first time several RNA vaccines has been developed and used and proven to be efficacious against this virus. And I think these technical platforms would help pave for future vaccine development as well. I guess the technology has been sitting there for a long time, but it would need something like COVID to make a last push for us to accept it and use it. But I think every cloud has a silver lining, and maybe this one is the RNA vaccine works, and we have like another tool added to our toolbox for future pandemic responses. And also, I do see like some of these repurposed drugs to treat for COVID treatment. Since we don't really have official medication against COVID-19, people are using these well-established or FDA-approved drugs to treat the patients and to reduce their symptoms. I think many therapeutic antibodies has been developed not only based on the convalescent plasma, or people had COVID and recovered from COVID, but and I think some groups really dig deep into old inventory, into the convalescent plasma of SARS patient, and they have also discovered antibodies from this quote-unquote library in order to use these antibodies against the COVID. So, the repurpose is kind of like an old approach to use the old reagents to do new things, and I think that can substantially cut short the development time of novel therapeutics. Of course, in terms of future outbreaks, I would hope that we can learn from this pandemic, and we can learn to embrace new things and new ideas. But I think what's more importantly is going to be strengthen public education about emerging infectious diseases. I think I read a book called Spillover. In the book, kind of pointed out that because we're taking as much surface area of this planet as we can because of the expanding urbanization and the explosion of populations, spillovers are kind of inevitable. So maybe, I'm not saying that I want this to happen, but I just think we will have another, maybe, I wouldn't say pandemic, but we will have another outbreak. But I think it's a matter of, when instead of if. But I do sincerely hope that, with the experience, or, with this painful experience for COVID, the public will be more prepared, and also we'll have an open mind. So when the next thing hits, we will be, better prepared, both technologically and mentally to deal with emergent infectious diseases.
Yeah, definitely. And yeah, I think that's right. I do, I wouldn't be surprised if there was, like you say, maybe not another full pandemic, but certainly, other sort of serious infections that kind of spread in the coming years. Because we did have, a couple of near misses. It was things like, avian flu and swine flu that could have potentially turned into full global pandemics, but didn't. And, this was the one that did, basically. But yeah, there's definitely some positive things to take from it. I think there's some good kind of optimism there to kind of end on, that hopefully we can deal with these kind of things better in the future. Okay, Yining, so thank you very much for joining us today. That's been really interesting.
Hey, thank you.
And yeah, thanks for sharing your expertise with us. Do make sure to check out sinobiological.com if you want to learn more about the research they've been doing on COVID and in other areas, or if you want to know more about their antibody products and the other services that they offer. So a big thank you again there to Dr. Chen for sharing his expertise with us, and thank you for listening.